So this is the fifth and final lecture on Covid-19, and we're going to cover three different controversies. Firstly, the sensitivity and specificity of the various SARS-CoV-2 tests. We're then going to look at vaccines, different sorts of vaccines, different strategies, whether they're likely to work, and we're also going to cover the likely origin of the virus. So before we start, it's well worth clicking on this link here on the original PowerPoint version of the slides. Have a read through this article, read it start to finish, and then see what you think and see if this has swayed your opinion as to whether COVID testing is good or bad, and we'll discuss that at the end of the session. So firstly, what is sensitivity and what is specificity? And the article that I've asked you to read through talks about sensitivity and specificity, and this relates to these are terms that are used for all diagnostic tests, and they relate to whether we can differentiate between healthy and diseased individuals using a test. And sensitivity reflects the percent of positive cases that are detected by a test, whereas specificity reflects how many positive results are true positives or how many negative patients would get a positive result. And a really good diagnostic test is both highly sensitive, so it detects all of the patients with disease, and it has a high specificity in that it doesn't give people who are negative a positive result. And this can be shown here on this graph, showing number of people or number of responses along here, and then here is an increasing range of something. In this case, we could say it's viral levels in the patient's swabs. Okay, so in healthy people, this would be below a certain cutoff, and you could argue that that cutoff should be 0. If it's 0, you're healthy. If you're diseased, you've got some virus. However, it's not as simple as that. We know that a lot of people have got some virus, but they don't suffer from COVID-19 disease, but they might be carriers. And this creates difficulty because this test is not diagnosing people with the disease, it's also flagging up people who are non-diseased and have the ability to pass on the disease, and we don't know whether they should fall into the diseased or the healthy category. For a normal test, we look at false positives and false negatives. So we have this cutoff value here. This is our threshold for detecting virus. If you are healthy, but you are above that threshold, that would be classed as a false positive. If you are diseased and you are below that threshold, so come back as a negative result, but you are diseased, that would be a false negative. Now, for most diagnostic situations, it's fairly clear cut. You come into hospital with some disease, your blood markers are above a certain threshold, and that is fairly characteristic for disease. However, we're dealing with a screening situation here where the overwhelming majority of people who are tested are negative, and there's only a small number of positives, and this can create a problem, especially because it creates a problem if your sensitivity and specificity are maybe near 99%, which sounds good, but if the rate of disease or the rate of positivity in your population is only 1%, then that creates a problem. So if the false positive rate is 1 in 100 and the frequency of a positive patient in a population is 1 in 100, then you test 100 people, you maybe find 2 positives, one's a true positive, one's a false positive. And this is what the article by Hennigan talks about. And this article by Hennigan is mathematically correct. It might seem a bit alarming, but it is mathematically correct. Where the article is not correct is it makes some assumptions and hypothetical situations which are not correct, and they don't represent the current COVID situation. So in the article it talks about, imagine if it's the false positive rate is 0.1% and the rate in the population is 0.1%. Well, those are assumptions, but they do not represent the current or even any of the previous situations. And it does ask the question, what is the false positive rate of the test? And there's some really interesting, very new data coming out talking about the positivity rate of the test. One of the major criticisms of the current COVID PCR test is nobody knows what the actual false positive rate is. The problem is when you create a new test, you need to find the false positive rate. If you're doing screening of hundreds of thousands of samples a day, where you're going to find 100,000 truly negative samples, and those samples just don't exist. So actually getting the false positive rate is not impossible, but very difficult to do, and we'll discuss ways of getting that data on the next slide. So this is just taking you through some of the arguments in the article that I linked to. So you have 10,000 people. Let's say you have 10,000 people who have a test. The false positive rate is 0.1%, and the prevalence in the population is 1 in 1,000. This is a situation that Hennigan suggests. What you can do is do a simple calculation. 10,000 times 0.1 over 100, so that's this 0.1% here, gives you 10 false positives. If your false positive rate is 0.1% and you do 10,000 tests, you're going to pick up 10 false positives. If the rate in the population is 1 in 1,000 and you do 10,000 tests, you're going to get 10 true positives. So in this situation, this mathematical scenario that Hennigan stated, it is true that you would detect 10 false positives and 10 true positives, so you would pick up as many false positives as true positives, which in the article he suggested that this was concerning. What I really find annoying about this article is it doesn't make any attempt to find out what the true false positive rate is, and there are various ways of doing that. You could look at data sets that have been published, such as in Ireland, which had a very low positivity rate for a period of a week. They did 187,000 tests, they got only 12 positives, which gives a positivity rate of 0.0064%. Now you could assume that every single one of those is a false positive, and therefore the false positive rate is this or lower. It's very doubtful that all 12 are false positives, but if they were, this would be the maximum possible false positive rate. Similarly, Wuhan, they tested 11 million citizens over the course of a week and got a positivity rate of 0.00028%. So this is also another estimate of the false positive rate if you assume that all 300 are false positives, which again is highly unlikely. The current UK best estimate is 0.0003%, and this is partly based on this data from Wuhan, and we use a slightly different testing system. They would, for complicated reasons, they would always have a lower false positive rate than us because they double test in a way that we don't. But this is a reasonable estimate. But let's just look at the Ireland data and see how that data stands up. So using that Irish data, do 10,000 tests, you've got a positivity rate of 0.0064 over 100. That would basically give you 0.64 positive tests for every 10,000 tests that are done. So even if the prevalence is low, 1 in 1,000, so if you do 10,000 tests and your prevalence rate is 1 in 1,000, you'd expect to get 10 positives and less than 1 false positives. So the outcome of this is, assuming that all 12 of these positives were false positives, which is highly unlikely, a positive result is 15 times more likely to be a true positive than a false positive. Basically saying all of this concern that Hennigan was raising in his article was, I won't say completely false concern, but it was not really making any attempt to get any true realistic data for these estimates that he stated. If we go on the best estimate of our current false positive rate of 0.0003% and we do 1 million tests per week, which we have been doing fairly recently, you do 1 million tests a week, with that false positive rate you would get 3 false positives. If the prevalence was 1 in 1,000, which it was a few weeks ago, you'd have 1,000 positives and 3 false positives. If the prevalence is 1 in 100, which it probably is by the time you listen to this lecture, there'd be 10,000 positives and 3 false positives. So what we've seen is, as the prevalence of the disease becomes more frequent, the false positive issue becomes far less of a problem. Now, one argument that Hennigan made over the summer when rates were low is how many of these positives are due to remnant virus. And this can be where people have had COVID-19 disease, they've recovered from that, but within their lungs is infected material. There's no viable virus anymore, but they are coughing up damaged bits of lung with bits of virus in there. And we know that people can remain positive for up to 80 days in extreme situations. Lots of people will remain positive for 2 weeks after they've recovered from COVID, but then that drops off very, very quickly. So it could partly explain why cases never fell to zero after the first wave, but what it can't do is explain why rates increased in August, September and October. If it was down to remnant virus, which is what Hennigan was claiming in August, then rates would continue to go down. Now, to summarise, a qPCR, or the PCR tests as far as you're concerned, are highly sensitive, highly specific, and the primers that we use should only give a positive result if the viral genome is present and there's no contamination. We know that some patients with low levels of virus might not be clinically relevant, and these are a different sort of false positive to a classic diagnostic test. They've got low levels of virus, but they might even not be infectious. So that is a potential false positive, but we're not really sure if they are true false positives. So what we think is happening the majority of the time is a test can be giving a genuine biochemical positive result because there is virus there, but the presence of virus is not being related to existing disease, it's relating to past disease. And in that case, you could argue that this is a clinical false positive. Now, the false positives are a real issue when we come on to antibody-based tests. And antibody-based tests antibodies detect either antigen, or we're detecting the presence of antibodies to the coronavirus in blood samples, as we covered last week. Now, the problem with antibody tests is there is no amplification step. In PCR, you amplify one copy of the viral genome to millions of copies. With antibody tests, there is no amplification step. So therefore, what you have to do is basically increase the amount of reagents that you use until you start to detect something. And this can then encourage non-specific binding of antibodies and cross-reactivity of what you think are Covid-19 antibodies to other coronaviruses which are not to do with this virus. So we think that with antibody tests, the false positivity rate is likely to be around 2%. And there's been commercial tests that you can buy at Boots and other pharmacies to test for antibodies, and the false positive rate in them is a few percent, and the sensitivity might only be 50%. So these antibody tests are not as good as the PCR tests for current infections. They're certainly not as good for as PCR for current infections compared to antigen tests. Antigen tests are not as sensitive and not as specific as the PCR tests. Okay, so in the live session we will discuss sensitivity and specificity and see if that Hennigan article changed anybody's opinions on how good various tests are. But we'll discuss that in detail in the session in the live Zoom session. So the next controversy, will vaccines work against this virus? And the honest answer is, we don't know. They probably will for a bit. I'll explain why I'm coming to that conclusion. We know that short-term IgG levels are maintained for 6 months or so, or we think that, and there's been a report out this morning suggesting that antibody positivity, so that's presence of antibodies in patients who had COVID in the spring, has dropped over the past few months, suggesting that detectable antibody levels will drop within a few months, and therefore immunity might diminish over the course of 6 months or so. We're now beginning to hear of many more patients infected twice within a 6-month period, which says that these are not very, very rare immunodeficient people. These are becoming more and more commonly found. And we're going to expect to see more and more of this as we go into next year. Now, if there was no immunity, we would expect hundreds of thousands of reinfections by now. We haven't seen that. We've seen small numbers of reinfections where people were infected very early on and then are picking up another infection about 6 months later. So we've certainly got short-term immunity in the vast majority of people. Whether that's long-term immunity is the big question. So one important thing to consider is what happens with other coronaviruses. There are four main coronaviruses that cause the common cold. 3 of them are shown here, and these graphs show the percent of patients over several years presenting with these coronaviruses with just common cold, and these are from routine screening type samples. And what it shows is that if you look at this one, what you find is you get very high prevalence in one year and very low prevalence in the next year, and then this seems to repeat itself in a roughly 2-year cycle or thereabouts. Other coronaviruses do something similar: high, low, high, low, high, low, and this one, it goes up and down year by year. Now what this suggests is if you've got peaks every 2 or 3 years, it suggests that the bulk of the population might be infected with the coronavirus, and then the following year the coronavirus is much less frequent due to some degree of herd immunity, and then a year later or 2 years later it comes back because that population immunity is lost. And if this is what's happening for 3 other coronaviruses, well, why should this particular virus be any different? And given what we know about people getting reinfected within 6 months, this may well be the pattern that follows for this particular virus as well. Now, if that is the case, protective herd immunity long term is not a strategy that's going to help completely suppress the virus. It might suppress it for a year and then it might come back, but annual vaccinations might work. So this is not a nail in the coffin for vaccines. It's saying that a single shot, like you would do for some diseases, is not going to work, but repeat vaccinations every year might work. And then there might be a point where you don't need that. Now, one thing that's really interesting here is there is some suggestion that the big flu of 1889, it was called the Russian flu, there's some suggestion that that might have been caused by this coronavirus. And the really interesting thing about this flu pandemic is it didn't come in a single year. It seemed to come back year after year. And I've not got any slides on this because I've literally only just found out this information. But if you look at the death figures for lots of countries that suffered from this, they tend to be a real spike in 1889 and 1890, and then it dropped off, and then another spike the year after, and then it dropped off, and then a year after that, you got another spike. And the data I've just been looking at was Swedish death records from 1889 to 1895, and there were 3 years where you got extremely high death rates. Then the following year it seemed to be lower, and then high, and then low. So it was over a period of 5 winters that the Russian flu really affected this country. Now, if that is the OC43 coronavirus, and there's a few groups now suggesting that is the case, that might be a good example of how this pandemic might pan out, is that you have peaks in 1 year, you get a bit of herd immunity, the next year there's diminished disease, the following year it then comes back with a immunity lasting a year or 2 years. So that's some interesting information that might be relevant, but then also you consider that if you vaccinate every single year, you may well be able to completely prevent this disease being a significant disease. So assuming that vaccination works, it's a good idea to look at what the current vaccine types are and what experience we have in vaccinating against coronaviruses. And at the moment there is no vaccine for MERS or SARS, and these are highly lethal diseases. But the reason why they have no vaccines is because both of those diseases were managed by controlling the population. It just happened to be that they weren't as contagious as this one. So we don't have any human coronavirus vaccines that work. None of those common cold coronaviruses have vaccines against them because it's not been financially viable to do it because they don't cause severe enough disease. What we do have is experience in veterinary medicine, so these are both alpha and beta coronaviruses. This one not that dissimilar from the one that we're dealing with here, same family. We know that we can get vaccines that work against these diseases, but you're vaccinating every year. Feline infectious peritonitis, lots of people will know because they'll have had their cats vaccinated for that one. That's an alpha. However, these are mostly gastrointestinal diseases, not lung diseases. So Maybe lung diseases are more difficult to vaccinate against. There is some suggestion about that. But what it proves is that vaccination to coronaviruses is possible, if not a bit short term. So there's a link here to some various coronaviruses that are in trials, and I'm going to go through the main types of vaccines that are in human trials or proposed to be in human trials, and talk a little bit about how they work. So the most common type of, well, one of the common types of vaccine are based around replication incompetent viruses, or viruses which have got reduced pathogenicity in humans. And anyone who's had the live flu vaccine, this is a strain of influenza that doesn't cause disease in humans, that has been engineered to express the current surface proteins of whatever the current H1N1 strain of flu is that's going around at the moment. So every year a new strain of flu pops up, and we create new vaccines that have the surface proteins for that particular strain. So we can just use a virus that's non-pathogenic to humans, genetically modify it to express those proteins, and then inject it into patients. Now what we're doing here, we're actually taking an adenovirus. This is a different family of virus to coronavirus. We have lots of strains which are replication incompetent, that is, they can't divide in human cells. Adenovirus is another type of virus that can cause the common cold, along with rhinovirus and other coronaviruses. So we can take this strain of replication incompetent adenovirus, engineer that strain to express the SARS-CoV-2 spike protein on its surface, and then inject that into people. The virus infects cells, but it doesn't replicate within cells, but the immune system recognizes that the virus is there. Cells become infected, they express viral proteins on their cell surface, and that creates an immune response. And if you click on this video, you'll get a video of the technology about how it's done. And if you click on this, you'll actually see one of the very difficult to read science papers showing that showing the first study of this virus, this adenovirus system in macaques, primate model. And this technology is very similar to the Oxford AstraZeneca vaccine that the UK government is pinning a lot of its hopes on. So this video is to a different company, but it's the same sort of technology used by the Oxford group. And this is a fairly standard vaccine strategy. So the next most common type of vaccine is the inactivated virus. So what we do here is we take the virus cultured in cells. In this case, we just use these cells called Vero cells, African green monkey kidney cells for some reason. They're just very good cells for growing viruses in. And so we culture the virus in these cells, isolate the virus, and then inactivate the virus. The inactivated virus is then mixed with aluminium salts and acts as what we call an adjuvant, which enhances the immune response. And this, I've just copied out the actual method of how it's done. It's very simple, or seems to be very simple. This is the method that the research group used to generate this vaccine. So you just take a strain of the virus that's taken from one of the patients in China, in this case, one of the Wuhan patients. Virus is cultured in Vero cells. Supernatant is inactivated with this beta propiolactone. It's just a chemical that disables the virus and kills the virus. Then you centrifuge that down to get rid of the cell debris, but you get the purified virus. And then you repeat the step again with this beta propiolactone. And then you mix it with 0.5 milligrams of alum, and then you pack it into syringes. So it seemed like a fairly simple, straightforward technology to create this vaccine. If you look back in the old literature on vaccinations, there are various other adjuvants. One of the old ones that was used is Freund's adjuvant, which is using some mycobacterium that you mix in with the vaccine to create an enhanced immune response. So you can put things in with the virus proteins that have been inactivated with something that will activate your immune system, and that potentiates the immune response. So this is fairly standard vaccine technology. So the next one is the Novovac spike protein nanoparticles. This is a completely new, a very new viral technology, and on the original PowerPoint version of the slides, just click on this point here and that will play the video explaining how this works. And this basically uses the SARS-CoV-2 spike protein and genetically engineers it so that these spike proteins cluster together to create a virus-sized, multi-lobed particle that's about the size of a virus, and then these are injected into patients. And this link takes you to the original study explaining the clinical trials. Okay, so you click here and the video will play on the original PowerPoint version of the slides. Okay, this is the final vaccine type, and this is an mRNA vaccine. And this is a completely new technology which has never been used for an approved licensed vaccine before. And it basically takes the viral mRNA encoding the spike protein, encases that protein, that isolated protein, within a lipid coat, and then you inject that into the patients, and the lipid allows, this lipid coat allows the mRNA to enter the human cells. The mRNA is then translated by a ribosome that produces spike proteins which are expressed on the surface of the host cell, and then that is picked up by macrophages and other antigen presenting cells, which then flag that up as a foreign protein. So if you want to see a bit more, try clicking on this link here. That tells you a little bit more about the technology. But basically we are artificially expressing the viral protein in our own cells, and that is a foreign protein, and then that will be flagging up to the immune system that this shouldn't. This is a foreign protein, and hopefully generate an immune response against that. Okay, so that's 4 different vaccine types. There might be other vaccine types out there. There is certainly some based upon a stripped down version of coronaviruses themselves rather than using adenovirus. But those are the 4 main ones that you're likely to see coming onto the market. Okay, I've just put this slide in because I found this, I mentioned this last week, but I thought it was quite interesting. And it's taken from this particular article here. And it's asked the question, does the route of infection matter? And we know that this virus infects lungs and causes pneumonia, but if you inoculate hamsters, which are a decent model of this virus, because the virus happily infects lots of different mammals, if you infect hamsters orally, then they will become virus positive, they'll have virus shedding, they'll be infectious, but they have no symptoms of pneumonia and no weight loss, little or no infection of the lung, and these animals do not die. If you give the virus to them by the respiratory routes and make them inhale the virus, they get very high viral load in the lungs, low viral load in the small intestines, in the same as here. They lose body weight, they show signs of pneumonia, signs of disease, sometimes die, and they show shedding of disease in both oral cavity and feces, as do these animals. So this is a really interesting thought, is can people be inoculated with the SARS-CoV-2 virus via a different route to effectively immunize them against pneumonia, but allow them to become infection positive? They'll probably get gastrointestinal tract disorder and diarrhea and so on, but they may not develop lethal pneumonia. So this is actually a possible thought that some people are considering, as can we infect people by a different route? And this is not without precedent. We know that with smallpox, you used to be able to take a smallpox scab, create a cut on the skin and tuck that scab under the patient's skin and seal that up, and the virus didn't replicate very well in that cut, and that gave the body the chance to develop an immune response against it before you got systemic disease. So this idea of vaccinating by infecting people deliberately via a different route to what is lethal is a distinct possibility. So just an interesting thing to think about. And this also might explain why some of the first people to die of COVID-19 in China were oral maxillofacial surgeons, basically people who operate on the nose and throat, and dentists as well. They were exposed to very, very high levels of virus that they were probably inhaling, and so when those doctors started dying, also ophthalmologists who would have received a large dose if they were examining someone's eyes and got coughed in the face, which is not a nice thought, but they were also at very high risk. So it is possible that an inhaled dose is far more lethal than a oral dose. Okay, I'm going to go on to the final controversy now, and this is the one that had most people talking early on in this year, is what is the origin of this virus? And I can put forward lots of suggested origins to this virus, but we don't have any direct evidence to any of them. It's all speculation. So I'm not saying this is where the virus came from. I'm giving you ideas as to where it could have come from and giving you what is currently known. So we'll stick with the facts. Firstly, in 2013, researchers at the Wuhan Institute of Virology isolate the bat coronavirus, specifically RATG13 is a name that's given to one of the many strains that they routinely grew and cultured at the Wuhan Institute of Virology. That is not in doubt. This group has published dozens of papers from this lab, and that's all verifiable. Second important fact to know, in March 2019, researchers at Guangzhou isolate a pangolin coronavirus from an imported Malayan pangolin. And importantly, look at the timeline, they published this paper in 2019, so this was the first ever pangolin coronavirus. They published that study in 2019, and in July 2020 had published a paper showing that they had managed to culture this virus. This was not at the Wuhan labs, this was in Guangzhou province. It says see article on next slide. I think that article's at the end of the lecture slides. November 2019, there are cases of respiratory disease both in Wuhan and Guangdong province, which contains the city of Guangzhou. Now this is the really interesting bit. The closest virus known to SARS-CoV-2 is the RATG13 bat coronavirus. So this virus here is the nearest thing to COVID-19 virus. However, it's not identical to it, and the virus also has parts which match directly to the pangolin coronavirus. And this pangolin coronavirus has the bit on it which allows entry into human cells, which is a version of the spike protein that can interact with human angiotensin converting enzyme 2, which we sort of talked about in week 1. So this bat virus can't enter human cells very well. This pangolin virus can. And what we find is that the SARS-CoV-2 virus is mostly matching very similar to this virus, but with a small bit of this virus that allows entry. So it's almost as if some host has been infected with both this virus and this virus at the same time, and that has allowed this SARS-CoV-2 virus to be so virulent. Now what's really interesting is there were 3 different types of this SARS coronavirus 2, and the one that's not really talked about very much is type A. And type A was found in Guangdong, where the pangolins were. Okay, so this has this is what we call the most primitive virus, which is the one that's effectively most similar to the pangolin and the bat virus. That was found in patients in Guangdong sometime at the end of 2019 and early 2020. Now around the same time, possibly earlier, and the timelines here are a bit strange, a bit unclear, is we know that in November type B virus was infecting patients in Wuhan. So there is some suggestion that the SARS-CoV-2 virus may have started in Guangdong because that seems to be a more primitive virus, as in if you genetically sequence that virus, the type A virus, you get the whole sequence, 30,000 bases of it, but then some changes have happened subsequently in the type B virus that make it more distinct from this bat virus. So it seems to be this bat virus is the closest, with a bit of pangolin in there. Then you go to type A, and then a few more changes have happened, you go to type B, and then another change has happened to create type C, and type C is what's infecting Europe and the West at the moment, and this contains the D614G mutation. So this is a change of the amino acid. The amino acid 614, so basically the 614th amino acid in the spike protein in the original virus and in presumably either the pangolin or the bat virus is aspartic acid. So the amino acid code D is aspartic acid. So amino acid 614 is aspartic acid, certainly in type B virus. Subsequently, a change has happened and changed that into a G, which is a glycine. So a genetic change has happened. We've gone from aspartic acid to glycine, and this is the virus that's affecting Europe and most of the Western world. And now, actually, to be honest, most of the world. This is much more infectious than the type B virus. The type A virus seems to have gone. We don't really know why. That might have not been particularly infectious, and this sometimes happens. If type A was the first one, that might not have been all that infectious, but then a genetic change happens to create type B, and then a genetic change happens to create type C, and then this is the one that takes off around the world. So we might be dealing with a more difficult to handle virus than China had it in the first place. So to summarise, you can do these plots, and these plots, the length of these lines represent genetic distance in a way. The bat virus is 96% identical to SARS-CoV-2, so it's different at 1200 out of 30,000 bases. So this is a big long line. It's out on a limb. So this is genetically distinct from type A virus. Type A virus is the closest thing to the bat virus, but then type A virus seems to have mutated to form type B virus, which really badly infected Wuhan, and then type B virus mutated to form type C virus, and that's what went off to infect Europe in January and February. So on all of these little spiky diagrams, long branches are large numbers of genetic changes, small branches are small genetic changes, small number of genetic changes. So it's only one very small genetic change between B and C to make it from type B virus to type C virus. So this is just a map of where we're dealing with. Yunnan is where the original bat virus was isolated from a cave. Guangdong and Guangzhou is down here, and Wuhan is up here. And a bit more on the type A virus. If you follow this link, this is a paper in a very big prestigious journal, the Proceedings of the National Academy of Sciences of America. So if you click on that link, you'll see a commentary on this where someone sequenced lots of very early coronaviruses from China, or took the sequences, analysed them, and found that early on in the outbreak, 7 of the 11 isolates were type A in Guangdong, which is 500 miles away from Wuhan. So there's something linking these two places together. So these authors are suggesting that the outbreak didn't originate in Wuhan, it could have originated in Guangdong. So the question is, did it originate because of the infected pangolins, which infected some people? You know, we know that pangolins are routinely eaten, sometimes eaten in restaurants, sometimes the blood is drunk raw, you know, so there were plenty of opportunities for the pangolin virus to infect humans, and then somehow the bat virus, either the one from the Wuhan labs or one that is naturally circulating, has infected the same host. So this is what this paper discusses in detail, which is where this quote is from, which can be summarised by this little map. 2013, the bat virus is first isolated, but this bat virus is still there. So there's many possibilities that any humans could come into contact with this virus. The pangolins we know were imported in March 2019. They were infected with the pangolin coronaviruses. There's some link somehow we've got to get this bat virus in contact with these coronaviruses to have a genetic recombination event to create the new virus. We know that the SARS-CoV-2 type A virus was circulating in Guangzhou region sometime between November and January. We don't exactly know when, but it was early on. Then somehow we've got transmission to Wuhan, where we know that this bat virus is grown in the Wuhan labs. That's not disputed. What we don't know is how this virus came in contact with this virus and infected people. And there are lots of speculations about how that can happen. It could be, eating pangolin meat, eating other, bat meat or whatever. We just don't know how that has happened. There's some speculation it could have happened at those labs, because we know that this lab is growing this particular virus. There's no published article suggesting that that lab has ever grown the pangolin virus in their own labs. But one possibility is if they had grown that pangolin virus, it could have recombined in a cell culture experiment. But as I say, this is entirely speculation, and we don't know how the recombination occurred. All we do know is a small portion of the pangolin virus that's crucial for entry into human cells has been effectively recombined. The pangolin virus has recombined probably in the same cell as a cell infected with either this bat virus or a very similar wild strain of this virus. They've got into the same cell. The virus genome was probably broken apart and recombined to create this new SARS-CoV-2 virus. We don't know if it happened here. We don't know if it happened here. But what we do know is it then mutated again and left on a plane out of China to Europe. And that's the type C virus that we're currently dealing with. So we can further discuss any of these topics. And if you look on the next set of slides, we've got just supplementary info, just in case you're interested. Some bits of information I've put together on genetic homology, as in how similar to the various viruses are. This might be a little bit tough for you, we'll see. How could a recombination event occur? It's all speculative. And then some links to some of the key papers. This is a really interesting article. It talks about the pangolin trade, and it talks about the how pangolins are imported, treated, and so if you click on that link there, it's a really unpleasant article talking about the pangolin meat trade. There's various other articles that I've looked at and highlighted that I've taken this information from. It's all out there on PubMed. That's the original description of the Guangzhou group who identified the coronaviruses in pangolins in September 2019. So the timelines, we know that it was first identified in March and they published the paper in November 2019. This is the original study suggesting of the genetic recombination between pangolin and the bat virus. And that's probably enough. So those supplementary slides are purely if you are interested and want to locate the information that I've used to put this lecture together. Okay, that's probably enough for now. I think this lecture's long enough. Okay, see you next time.